In the name of Allah, who is the most gracious and most merciful. Assalamu alaikum students, myself Kandil Fatma. Now we discuss protein fraction by precipitation. Main points: introduction, general principles, method, precipitation, reactors. Now we will go through introduction. Protein precipitation is widely used in downstream processing of biological products in order to concentrate proteins and purify them from various contaminants. Example: in the biotechnology industry, protein precipitation is used to eliminate contaminants commonly contained in blood. The underlying mechanism of precipitation is to alter the solvent precipitation is to alter the solvation potential of solvent, more specifically by lowering the solubility of the solute by addition. Now we discuss general principles: repulsive electrostatic forces, attractive electrostatic forces, precipitate formation. The solubility of proteins in aqueous buffer depends on the distribution of hydrophilic and hydrophobic amino acid residues on the protein surface. Repulsive electrostatic forces. Repulsive electrostatic forces from when proteins are dissolved in an electrolyte solution. These repulsive forces between proteins prevent aggregation and facilitate dissolution. These are the structures of ordering of ions near charged residues on a protein surface. Hydration layer and repulsive forces. These are the protein interaction caused by disruption of hydration layer. Now we tell about precipitate formation. Protein precipitation formation occurs in stepwise. First, a precipitating agent is added and solution is steadily mixed. Mixing causes the precipitate and protein to collide. Second, protein undergo a nucleation phase where submicroscopic size protein aggregates are generated. Third, growth of these reach a critical size for a high and low shear field by colliding into each other. During the final step called aging in Asia field, the precipitate particles repeatedly collide and stick, then break apart until a stable mean particle size is reached, which is dependent upon individual proteins. Salting out. Salting out is the most common method used to precipitate a protein. Addition of a neutral salt such as ammonium sulfate compresses the solvation layer and increases protein interaction. Energetic involved in salting out. Salting out is the spontaneous process when the right concentration of salt is reached in solution. The hydrophobic patches on the protein surface generate highly ordered watershed. This results in ensemble decrease in enthalpy and a larger decrease in streams. Next, precipitation with miscible solvent. Addition of miscible solvents such as ethanol or methanol to a solution may cause protein in the solution to precipitate. The solution layer around the protein will decrease as organic solvents progressive. Little place is water from protein surface. Flocculation by polyelectrolytes. Alkanate carboxymethyl cellulose, polyacrylic acid, tannic acid, and polyphosphates can form extended network between protein molecules in solution. Polyvalent metallic ions. Metals can be used at low concentration to precipitate enzymes, send nucleic acids from solution. Polyvalent metal ions. Precipitation reactors. There are numerous industrial-scale reactors that can be used to precipitate large amounts of protein, such as we complain and DNA polymerases. from solution. Batch reactor. Batch reactor is at the simplest type of precipitation. Tubular reactor. In tubular reactor, feed protein solution and precipitated reagent are contacted. Continuous stirred tank reactor. CSTR can run at steady state. Thanks for watching this video.